And we have a pleasure to have here Daniel Maria Busiello as a speaker today for our seminar. And Daniel did his PhD in physics, in theoretical physics in University of Padua with Amos Maritain, investigating the property of non-equilibrium systems. In particular, he was focusing on entropy production in a stochastic system and on its thermodynamics interpretation. And recently, he moved to Lausanne, to EPFL, with Paulo de los Rios, when he's focusing on really interesting research. He's trying to understand if system of chemical physics and in general biochemical reaction can be interpreted with stochastic thermodynamics, and if in particular the quantity of entropy production can be a good observable to understand the fate of a chemical reaction, and also in a very wide perspective, the origin of life. So I'm really happy to have him here, that we met in Padua many times, and I leave the scene to him. Thanks. Daniel, please turn on your microphone.
Okay, now it's on. Can you hear me now?
Yes.
Okay, perfect.
Now perfect.
Okay. So thanks for the presentation, and I'm actually glad to be not physically there in Granada, but presenting this talk. So, I mean, the title is Non-Equilibrium Selection of Chemical States Driven by Dissipation Phenomena. It's a really general title. And I would like to start with a bit of motivation, why we decided to start investigating this problem. And as Matteo already told, the motivation was the origin of life. So I will start this is really a motivation, and I want to explain the origin of life in this context, but let's start focusing the problem from this point of view. So if we track back the properties of living system that we know, we can actually track back the properties up to the, actually down, you can scale it down, the properties to the smallest part of living system that contains all the information that we need. And it is a single cell. Single cell, actually, it is itself a really complex environment, and it comprises both proteins and DNA in this really simple picture. DNA stores information, makes self-replication possible, and DNA also needs protein to work, to have a working environment. And proteins actually are molecular machines, really generally, broadly speaking, that keep cell alive and need DNA to self-replicate. So the question is, which came first? And here actually the RNA world hypothesis take place. And this tells that actually the RNA maybe came first, because RNA can perform both functions. It can act as a protein, as a molecular machine, and it can also store some information about the system. And this is just an hypothesis, but you see that we are actually moving the question to another question. Now it's not which came first. We suppose that we know which came first. The question is how it is possible that this collection of RNA actually could be stable without decaying at equilibrium, and actually having the chance to form more complex systems up to actually the ones that we know nowadays. And this big question actually can be declinated in several smaller questions. And the first one is the present oxidative condition maybe was different at the dawn of life. This is a possibility. And because in this condition, actually RNA is not stable at equilibrium. Another possibility is that the emergence of the high energy metastable state is possible at equilibrium. This is a question because actually it seems that it is not. And moreover, when did the switching from equilibrium to non-equilibrium environment take place? Because actually we know that nowadays most of living organisms, I don't want to say every living organism, but most of them actually operate in non-equilibrium environment, non-equilibrium conditions. So we are going to propose a different scenario, and the scenario is actually that non-equilibrium can trigger the formation of some complex states operating against their equilibrium fate to decaying toward basic building blocks. This perspective actually started as an inspiration with the original life problem, but can be applied to any kind of chemical system that evolves toward a sort, that follows a sort of path, evolutionary path. In any sense, in sense of population dynamics, in sense of just chemical evolution, in sense of catalytic enzymes, or in sense of the original life. In the general perspective is that external sources of energy actually can drive some molecules towards some more complex states that are actually unstable or unreachable or really with a really low population in non-equilibrium condition, in equilibrium conditions, sorry. And in the perspective of original life, this picture actually, it is inserted in the idea of the presence of deep sea vents that trigger this non-equilibrium condition and then can favor the formation of complex structures. And actually can stabilize them, because this is the point. The complex structure exists even in equilibrium, but they are not stable or they are present with a real low population. Hence, now the question is, what is possible in non-equilibrium condition? Is that picture possible or not? I would like to start with a really simple system just to understand the picture, and also because these really simple systems actually hides a lot of interesting features. Let's start. This is a three-state system. We have three states, A, B, and C. A is the high energy state. B and C are the low energy states. And in this picture, B and C have the same energy. I decided to pick the same energy for B and C just because in this case we have a complete symmetry in equilibrium condition. If we let the system relax to equilibrium, B and C will be equally populated in both boxes and even in total. Even in B1 plus B2 is equally populated with respect to C1 and C2 at equilibrium. These two boxes are simple picture for a thermal gradient, which in our case is the external source of energy. And again, this is in the idea of these hydrothermal vents near the submarine volcanoes, and that can generate this strong temperature gradient. So this is the inspiration why we use a temperature gradient. And also because it's a really physical source of energy, it naturally modifies transition rate. So it seems to us a good idea to start with this source of energy, essentially. So this is the simple picture, three-state system in two boxes, one temperature T1, the other temperature T2, where T1 is higher than T2. And this system can diffuse, each state can diffuse between boxes with diffusion coefficient dA, dB, and dC, in principle different. So the equilibrium conditions are when there is no diffusion, obviously the system will relax to a local Boltzmann distribution in T1 and in T2. In this case, the two boxes are simply trivially disconnected, and so each system relaxes to an equilibrium condition. There is no thermal gradient to experience. Another equilibrium condition is actually the absence of thermal gradient, but the presence of diffusion. So in this case, we have all T are equal, and in this case, the system will relax to a Boltzmann distribution characterized by a global temperature, not a local one, because now we have a global temperature which is the same for T1 and T2. This is another trivial equilibrium condition for the system. In general, when we have both diffusion and thermal gradient, we have some non-equilibrium condition, and we can start writing our reaction diffusion system. In this case, it's just a master equation because we are working with discrete space and discrete states. And this is the master equation that we can write. Is the first part is just the chemical part, the K. You see my mouse, I think. So the first part is just the chemical part. And the second part is just the diffusion. It comprises the diffusion rate. This is not really a standard diffusion at this level, standard in the sense of the Fokker-Planck description, but this is a diffusion rate. It's just a master equation, discrete space description, but the idea is the same. We also took dA equal to dB and equal to dC just for sake of simplicity, and we explored also different diffusion coefficient, and I'll leave for after later on I will explain which kind of interesting effect can take place when diffusion are not the same, but still the message stays the same. So let's start characterizing the system, and let's start with transition rates. So we obviously have to start with some thermodynamic constraint, because the important thing here is that we are not just throwing random rates to the system. We want to explain biochemical systems that are thermodynamically consistent. The thermodynamical constraints in this case are just can be applied to the ratio of transition rates. So in this case, the transition rate going from A to B divided by the one for going to B to A is just this Boltzmann factor. And this means that actually the system will actually relax to a Boltzmann distribution in equilibrium condition. So, sorry. Okay, we are back to the slides. And another condition, a thermodynamically constraint, has to be applied to the rates from A to C and from C to A analogously, and the same stuff also for the one from C. And another condition is that the rate from C to A and divided by the rate from B to A actually are related by this different Boltzmann factor, the one highlighted in red, that actually contains information about the energetic barriers, because each transition actually is dictated by the rates. It is dictated by the energy difference, but also by the energy barrier. Energy barriers control essentially the speed of the rates. So when we have two different energy barriers, delta AB and delta EC, we have that the reaction from A to C is faster than the reaction from A to B in both boxes, because the barrier is lower, basically. And the energy symmetry between B and C is kinetically broken in this sense. So it's broken by the kinetics of the reaction. And the state, and we actually observe that the state with the lowest energy barrier, so which is the one that experiences faster reaction, will be the most populated one at steady state. So essentially, when we broke this kinetic symmetry and we push the system out of equilibrium, C will be the most populated state. When I talk about the most populated one, I always intend the population of C1 plus the population of C2, because actually we want to look at the species C in the whole system. So we have to consider both boxes. And C will end up being the most populated one. This is just a sketch of my previous statement. And we see that actually we are seeing this difference population through the logarithm of RCB. RCB is nothing but the stabilization energy of C relative to B. So it's population, the global population of C divided by the global population of B. Essentially, the logarithm of this. Another interesting feature actually is that in this system, it appears that it is present an optimal gradient to trigger the selection of C with respect to B. So if you want to frame this message in what I told before, we have that two states which are actually equally parabolic at equilibrium. In non-equilibrium condition, this symmetry is broken and C is favored with respect to B, and actually is more favored when we drive the system with an optimal gradient. And this optimal gradient can be analytically estimated just by some expansion on the rate. There are simple calculation, and I'm not reporting here just to not bore you with calculation, but it's a simple stuff, and it can be estimated somehow. It's not really a precise estimation, but an approximate one, and it works fairly well. So essentially, the message is that thermal energy is transformed into chemical energy. So we want to understand why the fastest state is the most populated one in this case, because actually at equilibrium, whether you are fast or slow does not mean anything for your population. Out of equilibrium, the kinetics is important, and we can identify the basic rules dictating this selection by looking at diffusive cycles. These sort of diffusive cycles are sketched here on screen. And again, we have our system. Again, we have the barrier. We have the thermal gradient in two boxes. And we see that we can identify the stationary fluxes between both states. And you can immediately see that all fluxes satisfy the Kirchhoff's law, essentially, because we are a steady state. And we can identify two kinds of fluxes. One is the fast one, the other is the slow one, according to their magnitude. The fast one actually runs only through A and C, while the slow one runs only through states A and B. So essentially, the population of B is dictated by the slow cycle, while the population of C is dictated by the fast cycle. This is a really simple picture. Actually, if you complicate a bit the system, the picture has to be complicated a bit, but the idea is basically the same. So the message is you have to understand how kinetic is coupled to the state. And in this case, it's really simple. There are just two cycles, and we can understand the selection of C in this simple way through these diffusive cycles.
Excuse me, one question, please.
Yes.
What happens if you connect C2 with B1? What is the physical meaning of just you connect C's and B's if are identical?
Connect C and B. C and B in this case are not connected in this simple system. So this is just A can go to B, A can go to C, but B and C cannot transform to each other.
But is there any physical intuition why is realistic, or it's just the model?
This is at this level just a model, because we choose the simplest one in which we have energy symmetry that we can break. But actually, it is also realistic to some extent. And afterwards, in this talk, later on, I will present an example that actually fit this model, in which B does not transform to C. So it's actually realistic in some sense, but it is also a model because it's simple. But again, if you connect B and C, the picture does not dramatically change.
Thanks.
Do I answer your question?
Yes, thanks.
Okay. So going along this way, actually, now we have to understand what is the role of dissipation here, because somehow we can understand it through this cycle, because they are dissipative cycles. They represent probability currents, which, as we all know, are at the basis of the maintenance of a state or an out of equilibrium steady state. But actually, we would like to have a method, a quantity, to have this relation between selection and dissipation. So the most natural quantity we thought of is the entropy production rate. The entropy production rate in this simple system can just be written as proportional to the energy difference between A and B, the thermal gradient, which actually is the energy source, and the global current of the system. Sorry, there was a typo here. Is A2B2 plus JA2B2. Actually, this is A2B2 plus JA2C2. But anyway, the entropy production is, in the simple case, can be explicitly written in terms of the energy and thermal gradient. And in some good limits, the selection coefficient, which is RCB, the one that I actually introduced before, population of C divided by population of B, can be written as 1 plus something which is proportional to the entropy production, divided by delta E, because we have to scale the unity, and the other factor actually takes into account the barriers and probability of occupation of the high energy states. This formula highlights somehow how dissipation triggers and shapes the selection in our system. There is a 1 plus because at equilibrium, RCB is obviously 1. Out of equilibrium, the entropy production is different from 0, and we have 1 plus something which is positive. So this is a really simple formula to connect dissipation and selection, and it has been derived in the limit of infinite diffusion and in the limit of really small gradient. I mean, up to the second order in delta T formally. We can also derive a similar formula, interestingly, when a time-dependent temperature is applied to the system. So let's imagine now. I didn't actually. I will not talk about this particular case in detail, but let's imagine the same system as before, 3 states that can have reaction between A and B and A and C with the same barriers. And instead of being connected, the 2 boxes being connected, it's just 1 box, but the temperature now changes with time in a time-dependent way, actually in a periodic way. So to let the system relax to a periodic steady state. And also in this case, in same limits, essentially, for fast periodic driving, when system is driven really fast between different temperatures, we can explore different temperatures really fast. The relation is basically identical. When S has to be replaced by the time average of the entropy production rate among the whole period. But the connection is basically the same.
Daniel, sorry, could you maybe just comment a bit on what is the entropy production and the thermodynamic interpretation that you're giving to that, or the foundation of it, on the meaning in general, because everybody maybe is not familiar with this quantity.
Yeah, sure. Sorry for having not given a proper introduction to this quantity. The entropy production that I used is known as the Schnackenberg entropy production. Is the standard quantification of dissipation in mass equation system. So this formula originates from the work of Schnackenberg, and when you study a chemical system and you identify fluxes and affinities, you end up with this formula of entropy production rates. So this is actually essentially flux times affinity. It's a standard formula for chemical systems of the quantification of the dissipation of energy in the system. And it ended up being exactly this formula, the first part, sum over Kij Pi minus Kji Pj, the log of Kj over Kji, when the system is in steady state, and it can be described using a master equation. So this is the original derivation of this formula, but actually it has also some physical meaning. It can be demonstrated, for example, that in some cases, physical cases of a system connected with the different heat baths, this formula actually goes back to the heat dissipation into the environment. So there is some physical sense, some physical meaning for this entropy production. And actually, at this level, I would like to stay with this description, because in the last part of the talk, I will derive again the entropy production in a different way, and I will also derive a possible generalization for this entropy production that actually can highlight better the physical sense of this quantity. Because actually here the question is, what is the physical sense of entropy production? And I can tell you that this quantifies the dissipation, the heat dissipation to the environment. For chemical systems, it is exactly equal to flux time affinities. In a thermodynamic sense, this quantity can also be derived from the bilinear form force time flux and so on. But actually, the question here is slightly different, and it is what is the best quantity to predict somehow the selection in our system. And it seems that end reproduction, to some extent, it is a good quantity. But it is not the best one, actually, and I will explain this later. But if I've been clear on this point, why we started using end reproduction, I will go on. We'll talk.
Yeah, thank you.
So this is just a sketch of the correlation between the selection and reproduction, a bit beyond the limit that I actually tell you about, the infinite diffusion limit and small gradient. But it's not particularly informative in the sense. It's just to say that, okay, end reproduction seems to be a good quantity to somehow predict the selection. Now we can a bit extend the system to, if you want, a more realistic description of a reaction diffusion system. Again, in this case, we have the reaction part, which is the one with the case which are actually transition rates, exactly as before. But now we have no more two boxes, but we have now a one-dimensional system with a gradient. And the system can diffuse in this gradient. And now the diffusion is a standard diffusion in terms of Fokker-Planck description, is the d times the second derivative of p. So this is a proper reaction diffusion system in a thermal gradient. The thermal gradient enters in the transition rates because they depend on T of x. So obviously, transition rate depends on x through the temperature. In the infinite diffusion limit, the system goes back to a three-state system, trivially, because the system essentially experiences the whole gradient at the same time, and somehow it averages over the whole temperature experienced, and we go back to the previous case. Nothing special. When we have a small gradient, in this case, we can perform a small gradient expansion, and we can derive the selection in this case, where for selection, I mean, again, the population of C, how much increase with respect to the population of B for a reaction diffusion model in this continuous fashion. And the interesting thing here is that, I mean, the message is the same. Again, C will be the most populated one. Again, we have diffusive cycles. Again, we can visualize this diffusive cycle in the one-dimensional space. But here the interesting point is that since the system now is continuous, the domain is continuous, a characteristic length scale naturally emerges for the system. And this length scale is this LS is the square root of D over KBA plus 2 KAB. Sorry, okay, I want just to unblock my mouse. Okay, now it's fine. This is the characteristic length scale for the system, and it's just length scale. It's just diffusion over the reaction. I don't know if you are familiar with the Dunn-Kohler number, but essentially this is the Dunn-Kohler number. Dunn-Kohler number quantifies the interplay between diffusion and reaction. And somehow this is a sort of Dunn-Kohler number for this system. I will report on Dunn-Kohler number later on more in detail when I will talk about the specific example in which this theoretical framework applies. Now, in this case, we can see that for a continuous domain, still there exists an optimal temperature gradient, and we can visualize the selection of PC over PB, and this selection is maximized at an optimal delta T. Obviously, it is 0 when the state is 0, because system is at equilibrium. It goes up like delta t squared. This can be derived using series expansion. And eventually, it actually relaxes again to 1 when thermal gradient is too strong. Because for a too strong thermal gradient, essentially the system experiences an infinite temperature throughout the whole domain, and PC converges again to PB asymptotically. So that's why we can explain the presence of this maximum. Here it's really simple, and it's really obvious that actually a maximum should appear in the system. Let's start now to complicate a bit the system, because at this point we just know that the energy symmetry at equilibrium can be broken by non-equilibrium condition. We have an idea of how this can be broken, which is the quantity that can quantify this breakage of this energy symmetry. And this quantity is a dissipative quantity. And this actually explains the selection, what I call by selection of state C, which I mean that C has a really higher population with respect to B, with respect again to their equilibrium fate. So that's why this kind of study go back to the problem that I exposed at the beginning of this talk. Let's start complicating a bit the model. Now we have a chain of reaction. And why studying a chain of reaction? There are some systems that actually behave like this, chemical ones. But the interesting stuff actually comes from the result that this chain can give us, because in a perspective or an original life perspective, it is it is not so strange to think about a sort of chain of reaction, a connected chain of reaction. There are some examples in which a chain of reaction actually explains the emergence of some basic constituent of the life as we all know. So that's why I started to study this simple chain of reaction. And again, we have a three-state system, and we have barriers. And in this case, we have a set of high energy states, A, and a set of low energy states, B and C. Some of them are class R, some other are class L, where by class I only mean that class R is identifies system in which the fast reaction is on the right branch, A to C. Class L identifies system whose faster reaction is on the left branch, from A to, in this case, from A to C1. And we use B as a reference state. It's just a convention. We can use any reference state we want. So now the question is, obviously, which is the fastest? In the previous case, obviously, again, this whole chain of reaction can be driven time periodically out of equilibrium, or can be connected to another box, or can diffuse in space, independently of actually how you put the system out of equilibrium. The question is the same, which is the fastest in this case. In the previous case, we know that the fastest was C. So, okay, C is selected. But what about this case? Maybe C1 is the fastest because it's fast here and fast here. Or maybe C3 is the fastest because it is at the end of the chain, it is faster. Or maybe just C1, again, because it is faster in the absolute sense. It is not easy to answer this question just by looking at the system, but there is actually a procedure to answer this question. And again, the procedure resides and is rooted on stochastic thermodynamics. In this case, what dominates in the system is the fastest relative path, which means consider our reference state, in this case B, consider the path, I go back just for simplicity, consider the path from B to C2, and consider the path from C2 to B. So you go forward and then you go back. You evaluate the product of rates along this path and the product of rates along this other path. You divide them, and this is the relative weight of this path. So, and this is the quantity that actually dominates. This quantity has a physical sense, because this quantity is nothing but the entropy production, again, associated to this chain of reaction that bring a state in B to, that brings a particle that is in B to the state C2 in this case. So it is exactly the entropy production associated to the single path of a particle going from B to C2. And again, we see that this quantity dominates and can somehow give us a hint about the selection. In fact, I sketched here a representative realization of this system in which we have 1,000 possible states in this chain, and we see that slow and fast state in the absolute sense, so that which they are slow and fast in their own subsystem. In this case, they are completely mixed altogether. So we can find slow state selected and fast state non-selected, because it is the fastest relative path that actually dominates. This result can be also actually derived analytically in the infinite diffusion limit. Another interesting point is that in this kind of chain of reaction, there are some conditions where the one or the class R or the class L that dominates in the system. This means that actually all fast reaction are on the right branch or on the left branch, or almost all reaction. And in this case, we can see an explosion of selection of the last state of this chain with respect to B. This is really interesting, because if we can imagine an actual chain of reaction, and I would like to spoil that actually we are working with a chemist on this kind of real chain of reaction. And if you can work with this chain of reaction, and you can devise it such that all the reactions are on one side, you can see really an explosion of selection. This explosion of selection can hint at the promise of finding a really abundant species that actually is not so abundant or really unfavored at equilibrium. And actually, this is in this small box here. P 0.75 means that actually 75% of subsystem have the faster reaction on the right branch. And in fact, we see a deviation on that direction, which is exponential. And we arrive up to 10 to the 3, which is in amazingly large selection. It's also interesting that we see some global localization phenomena, a sort of emerging patterns in the system. So we see this cluster of states that are selected, and a huge number of states that actually is not selected or less selected in this case. And actually, this is remarkably different from the phenomenon known as Turing pattern, but it is again a pattern in this case. We don't have nonlinearities in our system. It is a linear system. It's a master equation 1, and this sort of pattern is driven by non-equilibrium conditions. Let's pass now to an example, finally, in which we can apply this theory. So the example is the well-known Farano's conundrum, which is the question is the following: Dribose in general can transform into pyranos and furanos in general, and it can a chemical reaction bringing him to different forms of pyranos and different forms of furanos. But actually, only furanos is presented in both RNA and DNA chains. So actually, this is a really long-standing question, and the question is actually why? Why, if pyranos and furanos exist in principle for any chemical system which originates from ribose, why only furanos is present in RNA and DNA chains? Obviously, I will present not the solution that will hold once and for all to this conundrum, but for sure will present a possible solution that at least partially can explain this abundance, or partially contributed to this abundance at the dawn of life. And this explanation is again rooted in non-equilibrium conditions, and we want to understand if it's plausible that a selection of Furanos is due to thermal gradient at the dawn of life. And this thermal gradient again originates from these deep thermal vents near the submarine volcans.
Daniel, sorry, can you maybe just like explain a bit better what are these chemicals from like a more general biochemical or biological like context for people that are not familiar with this?
Yes, I'm going to do it next slide.
Sorry.
Essentially, this is diribose, and this has pyranos and furanos. So this is diribose is just this chain of hydrogen, a series of I mean, as a carbon chain that you can see on the screen is one is on the center of the screen, and essentially diribose was the origin of a lot of chemical chains, originated several ingredients that actually now are present in protists that we observe, as in this case Faranos. These forms are just possible transformation, actually possible closures of this diribose form, which is an open form, and it can close in different ways, and it can form these different shapes. Diribose is itself an interesting molecule because it enters in a lot of chemical reactions that are interesting from the perspective of the origin of life. I'm not going into details on this, also because I'm not an expert in all other reactions of diribose. But in this specific context, we are looking at the possible closures of this open molecule. And these are all the possible closures. And in particular, this is the closure that we are interested in. This is the closure that we observe in vivo. And these all are the possible closures that we observe in vitro. And this is the energy landscape of this kind of system, where just below you can see all the energies and all the entropic factors of this system. It's not really necessary to go into details at this level. I want just to report the energies and entropic factors, because a really important difference here is that also the entropic factors come into play, because these structures can be formed in several conformations. So we have also an entropic factors coming into play with respect to the previous simplified model.
Daniel, we cannot see well the table because there is like a window on Meet Google that say that you are thank you.
Sorry. So this is these are the energies of the system, and you can see that actually Pyranos has lower energies than Faranos, which is an high energy state. You can see it from this table. Actually, it's quite interesting that we just observe beta Faranos in living systems instead of maybe the lowest energy states, which actually are favorable at equilibrium or near equilibrium. And actually, this is the point where we started our work. In particular, I mean, this is known as ribose isomerization network in literature. See, it's sort of standard network for this transformation of this open molecule. We did some we did some experimental characterization of this system, and we did experiment at equilibrium, try to fit the model on the experiment using the Boltzmann expression, the standard one for chemical weights. So this is a thermodynamically consistent model in the sense that I explained before. So we have all thermodynamic constraints on the rates are respected. And we actually were able to fit these curves and find the all the energies reported here, all the entropic factors. We confirm that experimentally with this kind of experiments. And so we see that the population of Pyranos is much higher than the population of Faranos for a large set of temperatures. At some point, we see that a crossing appears even at equilibrium. And this crossing is due to the entropic factors. So we see a crossing, but we see this crossing at amazingly high temperatures, first of all. And second of all, this crossing is actually a tiny, this is a tiny inversion of population that actually is not fully convincing in the sense that actually this is the main source of selection. And this is just an equilibrium picture. Note this. So this selection is tiny and appears at really high temperatures. We can build a model, a non-equilibrium model, as before, for this kind of system. Again, as I said before, we have an high energy state, which is the Dribose, and we have the low energy state, which is the Faranos and the Pyranos. And as I said before, Faranos is a higher energy state than Pyranos. And here the entropic factors come into play here. This eta sigma are the entropic factors. They come into play in the transition rates, and they are responsible for this tiny inversion even at equilibrium. What we can show, it is in a detail of this model is that actually we merge alpha and beta Pyranos and Furanos in one single moiety, just for sake of simplicity, and actually this is without any loss of generality at this level, because they have roughly the same energy and the same entropic factors. Okay, this is what I already told. The factors modifies transition rates, and we now can see the selection of Faranos with respect to Pyranos in out of equilibrium condition, and we see that actually is, I don't know if I have a table of this. I forgot to put a table, but actually the selection of Faranos with respect to Pyranos is arrive up to 50 or 60%. Which is, a bit much higher than the one possible at equilibrium. And the most interesting part is that this selection can be obtained for really lower values of temperatures, which are compatible with temperatures that are experienced. They are thought to be realistic in this context of hydrothermal vents. So we have two advantages. We can boost the selection of Faranos, and we can also boost it far before the emergence of this inversion at equilibrium, in the sense of thermal gradient of temperature, which was another critical point of the inversion observed in equilibrium, because it was really an amazingly high temperatures. Here, again, the relevant parameter in this case is what I called before the down-color number, which is, in this case, this quantity, where mu is the average value for the transition rates, and KD is just the value for the diffusion rates, exactly as before. And this down-color number encapsulates the interplay between kinetic effects and chemical reaction, in chemical reaction, and the transport processes, the diffusion processes of both reactants and products of the reaction. In this case, the chemical species, basically. What we choose as selection parameter in this case is not trivially the population of Faranos divided by the one of Pyranos, because we also have the entropic factor. So we modified a bit this selection parameter so that it goes back to 1 at equilibrium. It is just the steady population of Faranos divided, which is this one, divided by the equilibrium population of Faranos, and the opposite for Pyranos. And this goes back to 1 at equilibrium just by construction. And it is higher than 1 when the selection overcomes the equilibrium prediction, overcomes the equilibrium selection also, when at equilibrium Faranos is tiny selected. And again, also in this case, since the model is the same as before, and reproduction captures somehow the role of dissipation in driving this kind of selection for small gradients. Again, we can ask ourselves if there is an optimal value of selection even in this case, and as you can expect, the answer is yes. There is an optimal value of selection. Here you can see different plots for different values of the down-color number, which is an interesting quantity in our system, and we see that maximal selection is appears at optimal gradients, and this value, the value of this gradient, is actually compatible to these thermal drops in the isothermal bands, where chemical reactants actually is thought to do some sort of interaction, chemical interaction. Let's now pass to the last small part of this talk. I'm not going to bore you any longer about this part. What is interesting now is actually we saw how a thermodynamically consistent model can single out the essential feature that underpin selection in this context. And now we want to pass to a more general picture to understand which is the best way to capture all the information about the system. Because actually we studied a simple model so far, and we derived that the entropy production is a nice quantity close to equilibrium somehow, so for small gradients. So what happened far from equilibrium? Is entropy production still a good quantity, or we have to understand which kind of derivative functional do we want to use? So at equilibrium, we all know that the equilibrium solution can be found by minimizing the entropy production, which is trivially zero at equilibrium. And moreover, we have also the relation between the Kullback-Leibler divergence and the entropy production. Kullback-Leibler divergence just tells you how much a distribution is far from a target distribution. Our target distribution is the equilibrium one. Our distribution is the distribution of the states of the system in time. So if we see how this Kullback-Leibler divergence, how this distance measure evolves, we can see that it evolves following minus the entropy production. So the entropy production somehow is a good functional to study a system relaxing toward equilibrium because it converges to its minima. And the system follows this minimization trajectory during evolution. Alternatively, we can also say that the equilibrium distribution can be found by minimizing the free energy. So this is the standard free energy picture in equilibrium condition. So at this level, entropy production has been introduced as a mathematical quantity which is minimum at equilibrium. And again, this entropy production is the one that I introduced before. It quantifies dissipation, and it is zero when the system satisfies detailed balance by construction. Let's go a bit beyond this picture and let's study non-equilibrium situation in general. So it is possible. The question is it possible to find some G such that the non-equilibrium, it's a non-equilibrium P, obviously. It can be found just by minimizing this G, and so that this G somehow controls also the evolution of the system towards a target distribution. And in our case, the target distribution is the non-equilibrium steady-state distribution. The question is, what is this G? And moreover, as we discussed before, what is the physical meaning of this G? And what is the best quantity to look at? Because actually, as Matteo just asked me before, what is the physical meaning of entropy production? And this is really important because we want to understand what is the physical ingredient underlying this selection. And now let's suppose that we can find this G. We have also to find what is the physical meaning of this G. Otherwise, it is just a mathematical quantity. We cannot learn much more than formulas from our system. Let's start. At this level, I just want to sketch a priori derivation of this functional. I will go very fast on this because it's a really ugly calculation, but I just want to pass the idea of deriving this functional. So this functional can be written as s dot minus something, because we know that at equilibrium, g has to be equal to s dot, because as we saw before, at equilibrium, g is exactly equal to s dot. And we also have another contribution. We put a minus sign, ideally here, just because the picture is can be thought as following. We want to minimize s dot with the constraint of maximizing some sustained currents. So we want to sustain some non-equilibrium steady currents, maximize that current, while minimizing a reproduction. So there is this trade-off between s dot and j. So that's why we put an ideal minus here. This is the standard expression and reproduction. This is exactly equal to what I shown before. This is the Schnakenberg expression. Jij is nothing but kp minus kp, what I wrote before, and this beta fi minus fj is just the logarithm of kij divided kji when we impose the thermodynamic constraint. So we can understand this entropy production as the time derivative of the system entropy production, which is just the Shannon entropy, minus sum of P log P, standard one, plus a contribution, which is nothing but the flux of the difference of free energy. So somehow the entropy production quantifies how and tells you how the system tends to distribute this difference of free energy throughout the system, so to reach equilibrium. And these fluxes are actually fluxes of difference of free energies. And this is a really intuitive and also physical picture of entropy production. It's not groundbreaking, this sort of writing entropy production, but it is a nice picture to understand what is entropy production, just applying thermodynamic constraints. So now the question is, what is this J? J should have a similar form, because J still is a flux. So it is a probability flux on each state times some other quantity, some sort of log of delta G. What is this delta G? Delta G now has the same role of the Kij divided Kji as in equilibrium. But in this case, it should be non-local for sure, because we are looking at a non-equilibrium system. And we know that non-equilibrium systems experience some non-local effects, and it also has to depend on the dissipative cycles. If our system is a tree-like system, we have no cycles. The system cannot dissipate. I'm always thinking about chemical systems. The system cannot dissipate because there are no loops, so there are no thermodynamic forces acting on the system. There are no cycles. There is no dissipation. So G should be non-local and depend on dissipative cycles. What is the form of this G? An idea could be to use the Jarzynski-like statistics to quantify this delta G. Because the delta G somehow has to weight this dissipation along each cycle, and it has to be weighted by the paths bringing to that cycle, contributing to that cycle. If we have in our system a really isolated cycle, obviously the dissipation in that cycle will contribute less than a really connected cycle at the center of our chemical system. So there is a way to quantify this, to weight this sort of dissipation through its cycle, and we use the Jarzynski-like statistics just because in a dynamical sense, let me first say what is delta Q. The delta Q that I put here is here. is just the dissipation in one cycle, which is defined, as I saw before, the product of the rates in one direction divided by the product of rates in the other direction. So paths without loops, again, contribute only to a given cycle. And moreover, we can quantify in a sort of dynamical sense. Each cycle is the contribution to each cycle is given by the trajectory that dissipates only through a given cycle. This kind of dynamical interpretations is a suggestion to use Jasinski-like statistics, and we can go on with calculation, not shown here, and we end up with a formula of this kind. Obviously, this is a cumulant expansion formula, where we see that this J has a contribution, which is the average dissipation, another contribution, which is the variance of dissipation. So in principle, there should be some system, some chemical system, in which the non-equilibrium selection is governed by a new functional, which is not the entropy production, but is the entropy production minus this J, where this J takes into account the average dissipation and the variance of dissipation. So in some system, maybe this is the dominant quantity. And it just means that the system, the selection is dominated by the average dissipation on all cycles. But maybe there are systems, there could be systems for which this quantity is negligible, maybe close to equilibrium, and this other quantity can be a dominant one. So in this case, it's not relevant how much on average the system dissipates through cycles, but what is the variance of this dissipation? If there are cycles dissipating really less, so cycle dissipating a lot of heat, actually this gives a giant contribution to the variance. At this level, this is really theoretic, but this is the function that we derived from an a priori derivation with these arguments, and actually this functional do the trick. So if we minimize S dot minus this J, we find the stationary solution, exactly. And can be also, this can be also demonstrated analytically, just by doing a lot of calculation on this functional. So I will summarize the results of this talk. I have some additional slides, and if you are interested, I can show you, for example, what happens when the diffusion coefficients in the twisted system are different. And just as a spoiler, we will see that thermophoresis appears in the system. Thermophoresis is the accumulation of particles on one side of the gradient. It could be the cold or the high one, the cold or the warm one, and this is just triggered by thermal gradient and different diffusivities. But if you are interested, I can show you, but I will stop here for the moment. So just summarizing, non-equilibrium condition hence can drive the spontaneous selection of fast states in chemical system, and as a side result, also global localization phenomena of a cluster of chemical states in this giant biochemical network, ideally, can appear as an interplay between dissipation paths and energy landscape. So actually, in this way, we can single out the most important quantities, and we say that there are the presence of non-equilibrium condition and allegedly entropy production. These observations are amenable to experimental validation. In particular, the Farano's conundrum do the job, and we found a possible solution to this conundrum in the onset on non-equilibrium conditions that don't know life that can boost the selection of Farano's out of equilibrium. And as in the final part, I just showed that, yes, entropy production is a good quantity close to equilibrium, but maybe this is not the best quantity to quantify dissipation. And unfortunately, the best quantity to look at is not so easy to understand. It's not so easy to write either, because it is a weighted average of dissipation through each cycle of the graph. Actually, this is somehow expected because entropy production is a local quantity, while non-equilibrium conditions and selection as a consequence is a non-local quantity. So there should be some non-local functional to be studied, and non-local quantities usually are complicated in this kind of context. So this is, I mean, more a question than an answer. And this functional is difficult to study, but it is correct in the sense that it gives you the right stationary solution, hence the right selection, exactly the right selection, and quantifies dissipation by construction, essentially. I would like to thank my collaborator for these works, Paolo de los Rios at EPFL, and Xiling Liang, which is a PhD student in our group, and also Professor Francesco Piazza at the University of Orléans, which is now moving to Florence. And the source for this talk are the one paper recently published on communication chemistry, on the whole framework of twisted system, and also on the appearance of thermophoresis in this really simple context. And another one is a natural communication that will appear soon on the Farano's conundrum, where you can find also a lot of chemical details about all other possibilities to explain this Farano's conundrum, and all the other possibilities in general to tackle the problem of origin of life, if you are interested. And thanks for your attention.